Welcome to the Chapter 9 podcast, where we're going to start off and do a little bit of discussion about cell respiration in general and get into some of the specifics, and then we'll wrap it up with a second podcast that I'll put up a bit later. So, this podcast is going to focus on cellular respiration, which is the process that all organisms, I'm going to make this clear, everybody does this, are going to go through, which is going to allow them to take bigger chemicals, bigger organic compounds, typically things like proteins, carbohydrates, lipids, and we'll break them down into smaller pieces, and eventually, in most cases, if it's aerobic respiration at least, we'll break them down ultimately into carbon dioxide, which we exhale. So if people talk about like burning fat, what they're really trying to get across is if you do aerobic respiration using fat, a lipid, you can actually break it down into CO2 and essentially exhale it. Now that does not mean that you can sit there and like hyperventilate and lose weight. This is only going to be if you actually have a purpose where you have to use it up. Just breathing out does not mean that you're really like losing weight in any meaningful way. And of course, when we break things down, what we're really trying to talk about is while we break all this apart into smaller and smaller pieces, we release energy, which we are going to trap and store as ATP. So this will be our guiding idea here of breaking big stuff down into smaller and smaller pieces, and in the process of breaking into these more energetically stable, lower energy level, smaller pieces, the energy that's released can be kind of coupled with the reaction where we build ATP from ADP plus an inorganic phosphate. And then that obviously can be used elsewhere once we have ATP for whatever we want. That's our energy currency. So that can go elsewhere and we can use it how we please. And then we've got autotrophs and we've got heterotrophs. And the idea here, as I said, everybody ultimately is going to do cell respiration. That's a universal process. But some organisms, beyond just doing cell respiration, can also do photosynthesis or chemosynthesis, where they ultimately make their food. That's the synthesis part. They also make their structural compounds, so they can make, cellulose, they can make enzymes and other proteins they need, nucleic acids. So don't assume that these guys, when we talk about a lot of times, that they make food because we think of plants and go, yes, we eat them. That's our main concern. They do build them for all the stuff that they need, for growth, for reproduction, for functioning. Only a small chunk of what they actually make will be stored, such as in like a potato or a carrot or something like that, where it's stored in the root system. And you'll see here that plants do this, of course. Algae do this, which are kind of like the eukaryotic, simpler version of plants. That's where plants evolve from, one subgroup of green algae. But there's lots of other types of algae that are everywhere, from unicellular to gigantic brown kelp forests that are huge. I mean, you're talking hundreds of feet tall in some cases. And then we've got the simplest guys, these cyanobacteria. And so these guys are prokaryotes, and they are the ancestors of all the photosynthetic guys, at least, because what happened was some of these cyanobacteria appear to have moved into, we call it endosymbiosis, moved into larger cells and ultimately gave rise to chloroplasts. So when you see eukaryotic cells like algae and plants that have these chloroplasts, you can kind of think of those chloroplasts as being what used to be endosymbionts of cyanobacteria, so little guys that were living inside of there, but they were kind of their own guys still. They were just, working together with the cell, that eventually got to the point that enough time passed that these cells started to get some control over the chloroplast as genes kind of migrated from the chloroplast, if you will, to the nucleus. And so the nucleus had some control over what happened with them. And so we no longer considered them regular bacteria now. We considered them to be an organelle, or the chloroplast that's inside of a eukaryotic cell. So these guys will be able to do photosynthesis. There are some guys that can also do chemosynthesis. There's other ways. Usually it's light, though. So they'll use light energy to take carbon dioxide and water, and then use that light energy to convert it to organic compounds, and they have oxygen as a byproduct. You'll then see that plants, algae, bacteria, and then all of the animals and fungi and other heterotrophs, everybody does this step where they can take organic compounds and oxygen and then break those organic compounds down using the oxygen to ultimately get carbon dioxide and water, which is kind of cool because this is the same thing that happens if you burn it. So if you took your food and just took a match to it and let it catch on fire, that's the same chemical process that's occurring overall when we're doing cell respiration. We obviously do it bit by bit to make sure that we don't, self-immolate. But in general, it's still the same end result, assuming that you're using oxygen, that it's aerobic. You'll see there's some other versions that are a bit simpler, that don't give you as much energy, that are called anaerobic, that don't need oxygen. But the one that we normally will talk about will be aerobic because it uses this oxygen. You'll also see that plants and these autotrophs in general, we oftentimes don't think of them as doing cell respiration. A lot of people just go, oh, they make their own food. But they still have to digest that food, if you will, because they still have to convert that food into ATP. So even plants, algae, bacteria, all these guys are still going to go and do this kind of complete cycle. The issue is because so much of what they make is either stored or so much of what they make is part of their structures. Like I said before, it's enzymes, it's phospholipids, it's the stuff they need to make more cells, to make their cells function. For them, a lot of the stuff that they ultimately make, they don't break down as food. Only a small portion of what they go through and make during photosynthesis is actually going to be digested. So in other words, they do photosynthesis a lot more than they do cell respiration. So as a result, we oftentimes think that there's this net gain because they do photosynthesis more, and some people try to ignore the fact that they do cell respiration, but that would be wrong. All of these guys will still do the cell respiration process. It's just only the autotrophs can do this process where they convert CO2 and water to organic compounds, and that's why we need them so badly as animals and heterotrophs. So terms, catabolic, this is just going to be that process of breaking things down into smaller pieces. So this typically is going to be what involves hydrolysis that we've talked about before. So cell respiration as a whole is predominantly going to be a catabolic pathway, where we're taking these large organic compounds and at the end typically ripping them apart into CO2s. You'll see some water is produced as well, but if we track the carbon at least, it's going from a sugar or a lipid that has lots and lots of carbon, and it's becoming CO2 where they're single carbon molecules. We've kind of torn them apart. Now we will have some processes that are building that would be endergonic or anabolic. Specifically, you'll see that during cell respiration, we're going to focus a lot on taking an inorganic phosphate, an ADP, and converting it to ATP, which is a building process. And so we're normally going to take this breaking apart idea and building, and we're going to use coupling, energy coupling, to allow us to use the energy released by this exergonic destruction to allow us to do this endergonic creation of ATP. Aerobic cellular respiration is what we'll talk about mostly, and this just means it's going to be with oxygen. There are other ways you can do this that's anaerobic, that does not need oxygen, but they're much less efficient, we'll see, on the order of about 2 ATP per glucose versus more on the order of about 36. So you'll see most organisms evolutionarily, for the sake of efficiency, are going to favor aerobic if they can. Obviously, some things have adapted to environments without oxygen, so they don't have much of a choice. But if possible, this is going to be the default pathway, aerobic, just because of that fact. And then this is an overall sample formula. When we talk about cell respiration, you can break down pretty much any organic compound. But our poster child here is going to be C6H12O6 or glucose. It's not like this is the only thing that we break down. It is a common thing we break down, but there's plenty of other stuff. It's just that's a lot of things to remember if I try to throw how to break down 30 different molecules. So we're going to focus on one and use that as our poster child or our example. And so you can see here we take glucose and then we take 6 oxygens. We react those together, and this will not be all at once. We're going to do it kind of bit by bit through a series of small reactions. It's kind of like making a waterfall be a bunch of small waterfalls. Instead, it still goes down the same distance, but it doesn't do it all at once. You get more like that staircase where most people would rather take the staircase versus just, falling down 10 feet. Overall, you end up on the ground, but there's a little bit of a difference as far as the safety of getting there. And so these things are going to react, and they're going to give us 6 CO2s, 6 H2O, and this energy. And the energy we talk about always in any type of cellular respiration is going to be ATP. That's what we're going to make. That's how we're going to store this energy for later. Okay, some quick terms, especially for those of you that haven't had AP chemistry or maybe didn't have as thorough when you were going through chemistry. These things will involve something called redox reactions, which just involve electrons changing hands. So in a redox reaction, you're going to have some type of electron movement from one substance to another. That's the general theme here. And to memorize this, you can think of oil rig. And the idea here is oxidation is losing So if you're talking about what's happening to a compound during a redox reaction, well, the guy that's undergoing oxidation is losing electrons, all right? Its electrons are being stripped away. When we talk about reduction, the rig part, reduction is gaining. So that means that the guy that's being reduced is ultimately going to gain electrons. So if we try to give kind of an example here, you could have where oxidation could make sodium, Na, become Na+. It's lost an electron. Whereas you could have where reduction could make something like calcium 2+ ultimately get converted to just calcium. Because to do this, calcium 2+ needs 2 electrons to become regular calcium. So it gained electrons. Whereas for sodium to go from regular sodium, sodium atom, to being a sodium ion, it ultimately had to lose 1 electron. Okay? It had to give 1 up. And then you might see these other terminologies where they say reducing agent or oxidizing agent. Because we're not saying you're being reduced, but you're allowing something else to be reduced, it's actually going to be the opposite. So reduction's gaining electrons, but a reducing agent donates electrons. It loses electrons, which allows somebody else to be reduced. To be reduced, you have to gain an electron, so it has to come from someone. So if you see them talk about reducing agents, just think that's the guy being oxidized. If you see them talk about a oxidizing agent, that's the guy being reduced. Because you're allowing, by being an oxidizing agent, you're allowing somebody to be oxidized, which means you have to be reduced to allow that to occur. Now, as we kind of discuss in practicality how this is going to work, your food is going to generate the vast majority of its energy by allowing electrons to kind of be stripped from it. I guess allowing, it's a chemical reaction, by having electrons stripped from it. And so these electrons that are stripped from the food are going to ultimately reduce a molecule called NAD+ to become NADH. And you'll see there's a common theme here where we're typically going to give H+ and electrons to these what we call electron carrier molecules. Right? There's technically 2. And the 2 that you'll see are NAD+ is the big one, and then you might also see FAD. And that one ultimately gets reduced to become FADH2. Now when we're looking at these guys, you're going to see that the H+ that we've talked about, that is ultimately going to be packaged up with electrons. And so what we're really going to end up doing is making this a hydride, like an H-, where we typically pack up an H+ with 2 electrons. And so dehydrogenase is going to be the enzyme that does this, where it takes this H-, which is essentially just an H+ plus 2 electrons, and it can use that to add it to someone like NAD+. If you take NAD+ and you add an H-, because that's really what we're talking about here, you get ultimately NADH, completely neutral. And so as we build these electron carriers, this dehydrogenase enzyme is going to take these hydrogens with these packaged electrons, and it's going to stick them onto guys like NAD+ or FAD to reduce them. These reduced molecules can then go to the electron transport chain, which we'll discuss later in more detail, but that will be able to then extract these energy-rich electrons from these reduced electron carriers, and then use the energy that they possess to generate ATP. Now this will only happen if you're doing aerobic respiration, because you need oxygen to do this kind of reverse process where we can take the electrons from NADH and kind of use them up, because they have to go somewhere, which allows us to convert NADH back to NAD+ in a safe and usable way. And so without oxygen, we can't really do this well. The electron transport chain clogs up and fails. And so that's why it's so essential that we have aerobic respiration with oxygen, so we can properly extract these energy-rich electrons from these reduced NADHs to oxidize them back to NAD+, so they can keep getting reused. You'll see that there's going to be a cycle here when we talk about these guys, where they kind of go back and forth, but it lets us use that energy then to make ATP. And that's where the vast majority of ATP that's going to come from cell respiration, or at least aerobic cell respiration, will come from, is this process using the electron transport chain and the electron's energy. You'll see eventually we actually use it to make an H+ gradient, and then that will generate a bunch of ATP, the vast majority. If you're doing anaerobic respiration, we can't really use these molecules, and so we end up just wasting them. It's kind of like having a specific food that might be wonderful, but you're allergic to it, so you pretty much just have to get rid of it because we can't use it properly. And so for anaerobic, these electron carriers become more of a liability because we can't properly use them. In their reduced state, we can't make anything out of them, so we end up just having to oxidize them back to NAD plus for nothing. Like, we don't get any value from them. There's no energy gain. There's no ATP gain. We've just squandered the energy that was in these very, very energy-rich molecules, NADH or FADH2. Electron transport chains, we're going to pick this up a little bit more later, but I want to go through it with this redox idea because it's kind of important. So the electron transport chain is going to be made up of these cytochrome proteins, so a series of proteins. It's going to be embedded in the cristae, which is the inner mitochondrial membrane. So if you draw mitochondria, there's normally that membrane inside that's drawn with these folds. That's the cristae. So these proteins will kind of be embedded in here. They'll just be kind of wedged. So as I try to kind of draw some examples, you'll see them in a row kind of embedded there. And they're going to allow electrons to kind of jump from one to the next to the next. And each time they do that, they go to a lower energy arrangement. And this allows for them to give off energy. And that energy is actually going to be used to pump H pluses, which once again we'll discuss a bit later, which will allow us to pump H pluses, which will allow us to make ATP. That's the goal of this, is to go through and at the end of this process, after we've used the electron transport chain to do things properly, we're going to be able to use this to generate a bunch of ATP. And the electrons that have been kind of ripped away from the food, they have to go somewhere at the end. So in this case, where they're going to go is they're going to go on oxygen. And specifically, they're going to go on oxygen, but they're also going to go with several of these H pluses that we have. And so it's kind of cool that once we add in these electrons, these H pluses, and the oxygen at the very end, we get water. So when they talk about like metabolic water, sometimes they'll mention things like kangaroo rats live in the desert and they get a lot of their water metabolically. It's because when they go through this electron transport chain process and they dump those electrons on oxygen at the end of the electron transport chain, it allows them to produce water. Now that water is not going to be a ridiculously high amount. Most of you guys have to drink a lot because it doesn't meet anywhere near your needs. But for someone who's very, very, very frugal about their water use that lives in a desert, for many of them it can be a fairly significant amount. So it's not always going to be something that we kind of scoff at or something that's trivial. But for us, we normally are going to get most of our water through drinking. So strip it from the food, move it to the electron carriers, move it to the electron transport chain, and then eventually it dumps on oxygen. Why are we doing all this? To make ATP. That's our overview as far as the redox idea. Okay, this is kind of an overview of the whole process. So I'm going to kind of try to get you guys to get a big picture, and then at some point I'll wrap up and we'll pick up with some of the other stuff later. But you'll see aerobic respiration is what we're dealing with. So anaerobic, we'll kind of talk about, that's what's over on the right here where it says fermentation. This is what you can do if you're going to go with anaerobic. You'll see it's labeled. So for aerobic respiration, in the cytoplasm, you're going to do glycolysis. Glycolysis is pretty much just going to kind of Chuck Norris split the 6-carbon glucose into 2 3-carbon pyruvates is what we'd call that. So we've split it in half, and in the process of this, we're going to generate 2 ATP, and we're going to generate 2 NADH. That's our ultimate end product that we've gained. So those 2 ATP could be used for energy. The 2 NADH can ultimately be saved, and they can eventually be plugged in later to the electron transport chain. So these guys would kind of migrate around, and we would use them later when we're doing the electron transport chain. If we did not have oxygen, at this point after glycolysis, we would convert the pyruvate, these 3-carbon compounds we got 2 of them by splitting a 6. Split a 6, you get 2 3s. We could ultimately get and convert the pyruvate into lactic acid or ethanol. Those are our 2 common fermentation pathways. So without any oxygen present, we would just do one more quick step, and this additional step you'll see does not actually give us any ATP. This is why it's kind of crappy, because all you're going to do is convert pyruvate to some other compound that you don't really need, but in the process you'll see here that we convert NAD plus back to NAD plus. Let me see if I said that correct. We'll convert NADH back into NAD plus because we need NAD plus to do glycolysis. So if we kept doing glycolysis too much, because glycolysis does not need oxygen, But if you keep doing it without oxygen present, where all you do is glycolysis, eventually all your NAD plus is converted to NADH and the process stops. And that's a bad thing. So fermentation, really all it's there to do is convert NADH back into NAD plus. And you don't get any energy from this. You're basically squandering it. It's just you have no choice. If you want to keep doing cell respiration, you have to get rid of this NADH. Even though it has a whole bunch of value, it has no value to us in this situation. It's kind of like having a whole bunch of American dollars if you're somewhere where they don't accept American dollars. And so even though it has potential value, we can't access it. And in some cases, if you don't have room for something, you're going to want to pitch the money that you can't use and save the money you can. And that's kind of like what you have going on here, is you want to keep making ATP, so you pitch this NADH because you can't use it. Now, if oxygen is present, we're going to go through and do aerobic, where we're going to move on into the center of the mitochondria, call that the matrix, and we're going to do a step that's oftentimes called the conversion step, where we convert pyruvate to acetyl-CoA. So it essentially goes from a 3-carbon compound to a 2-carbon compound. And then acetyl-CoA enters the Krebs cycle, sometimes called the citric acid cycle, or the TCA cycle, which is just the citric acid cycle. It'll go into this cycle here. And the goal of the Krebs cycle, if you will, is to take and break down this acetyl-CoA into CO2. And in the process, we generate a whole bunch of NADH, FADH2, and we get a little bit of ATP that we can ultimately make. There's about 2 of them. Here it's listed as GTP, but you can convert that to ATP. So we generate a little bit of ATP, but pretty much nil. The goal, if you will, of Krebs cycle is to get as many of those high energy electrons off the food and store them in those electron carrier molecules for the electron transport step, which is the next step, the last step. And so that's what we're trying to do, is just tear that food apart into CO2. And so that's the 1-carbon molecule you see here, is where we've ripped them apart into CO2s. But in the process, we've gotten all these electrons that can then move on over to the electron transport chain to provide us with a bunch of energy to make ATP. And so our last step is going to be the electron transport chain. It's oftentimes also referred to because as oxidative phosphorylation. Wow, that was horribly drawn. And the purpose for that is because when you're looking at oxidative phosphorylation, the goal there is that you are going to take and add phosphates to ATP. That's what we're going to consider phosphorylation. So if I mention phosphorylation at all, it's always going to be this process of ADP plus a phosphate to make ATP. So that's what I'm referring to with phosphorylation in this context. And so oxidative phosphorylation is going to use an H plus gradient to make ATP. So when you see oxidative phosphorylation, kind of draw an arrow, it's going to use an H plus gradient, and this is critical, to make ATP. That's how most of the ATP is going to be made when we're doing aerobic cellular respiration, is we're going to take the energy from these electrons. So we're taking the energy from NADH, from FADH2, and by ripping those electrons off, which you can see here where the NADH goes back to NAD+, the FADH2 goes back to FAD. So by ripping those electrons off and putting them through this transport chain, it allows us to pump H pluses into this, what we call the intermembrane space, or essentially out of the cristae. And then when we let the H plus diffuse through a specific protein, and you can see that protein right here, it's called ATP synthase. So by allowing it to diffuse back in, it basically spins this, I'm trying to think of the best term I have. If you're trying to like generate electricity, you have like a turbine. And so by letting the H plus through, it essentially spins a turbine that allows ATP synthase to make ATP. And so this whole process is trying to take the electron from the energy, take the energy from the electrons, use that to pump H plus out, and then let the H plus in to actually make the ATP. So it seems convoluted, but it's actually very, very efficient when it comes down to it. This will allow us to typically generate 34 ATP that we'll talk a bit more about later, but it makes 34 ATP out of a total 36 to 38 max, if I can write, max that we can make during aerobic cell respiration. So by far, this is going to be the most critical step, the most energetic step that we have in cell respiration, period. So I think we're ultimately going to stop there. Coming up after this, we're going to go through and discuss the steps a little bit more in depth. So we've kind of given our overview of the steps that are involved. Once again, we've got glycolysis, the conversion step, which I've asterisked just because a lot of times they skip over that and just kind of tuck it into the Krebs or the citric acid cycle. And so we've talked about citric acid cycle, the ETC, as well as the whole oxidative phosphorylation idea. So we're going to go through each of these in more depth in the next podcast to kind of discuss exact amounts, exactly what's going on, and where it's going on. So you've gotten quite a bit of the big picture already, but just keep in mind we will go through this in more detail to make sure you're comfortable with some of the general things of where, who's involved, and how much you get of everything.